the last class we have discussed DNA replication. You have learned the central dogma of molecular biology. Okay. See, soon after proposing the double helical model, Watson and Crick proposed semi-conservative model of DNA replication. The central dogma of molecular biology says that DNA, the informations in the DNA will be copied to an mRNA, to RNA. Okay. Then these informations can be given or encoded in the form of protein. Okay. That means this gives the fundamental of the gene expression. How is gene expressed? How DNA can express our character. A DNA will first form RNA. This RNA helps in the production of protein. Then you have already learned DNA can form DNA. Okay. DNA can double and produce new copies, and this process is known as formation of information from the DNA. If it is copied to DNA itself, we call it as replication. Okay, last class you have already learned it. DNA to DNA is replication. The next process, the informations in the DNA, if it is copied to RNA, that process is known as transcription. That is what we are going to discuss today. Okay, and see, when we discuss transcription, you should also know what is reverse transcription. If transcription is the forward thing, DNA to RNA, what is reverse transcription? If the information is in the RNA, it was copied to DNA, it was converted to DNA, I mean RNA to DNA, we can call it as what? Reverse transcription. Okay, it takes place in some viruses. Okay, in the case of human immunodeficiency virus, HIV, when HIV enters human body, its RNA should be reversely transcribed to DNA, and this DNA can make changes in our cells. Okay, so any virus with RNA as the genetic material, in order to produce its effect in the host cell, should undergo reverse transcription. I mean RNA to DNA conversion. I mean the synthesis is known as what? Reverse transcription. The last step, RNA to protein. I mean the informations in the RNA, if it is decoded or copied in the form of protein, that is known as translation. Okay, translation. Altogether, these processes determine our characteristics. So this is known as, I mean DNA to RNA, RNA to protein. This is known as central dogma of molecular biology. Okay, very simple concept. Then come to the first part, transcription. Okay, in detail. You have learned when a cell undergoes, its DNA undergoes replication, the entire DNA is replicated. Okay, the full DNA is what? Divided and replicated. You have learned it. Okay, DNA as such is replicated to produce two DNA, daughter DNA. Whereas for transcription, okay, you have learned that the chromatic material is classified to heterochromatin and euchromatin. And you have learned heterochromatin is not transcriptionally active, only euchromatin takes part in transcription. Means the loosely arranged regions, not the dense region. Only the euchromatin takes part in transcription. So understand, in the long DNA, only certain places are involved in transcription. I mean, if the DNA is of 2 meters long, only some regions of the DNA undergo transcription or involved in transcription. Okay, this is very less portion, less than 2% only. That we will discuss later. Anyway, transcription occurs only at certain sites of the DNA, not to the entire DNA. Okay, entire DNA is replicated, but only very selected regions of DNA is transcribed. Okay, before moving to that, understand when DNA replicates, okay, both the strands synthesize opposite strands. Is it not? Both the strands act as template and synthesize new DNA. Okay, this is old DNA and this is old DNA. Okay, this is new DNA. Okay, then both are DNA strands. Whereas, when the DNA forms RNA, only one strand is copied to RNA. Okay, when this particular DNA produces RNA, I mean when it is transcribed to RNA, single strand only getting transcribed. So that strand is known as template strand. Only the template strand is copied to RNA, not both the strands. Why it is so? Why both the DNA strands are not copied to RNA? Why only single strand or template strand is copied to RNA? There are two possible answers for that. Okay, both you have to learn. It was asked as a board exam question. Okay, so the first reason. Okay, this strand produces an RNA. I mean, the template strand produces an RNA. Okay, now what is the difference? If it's adenine here, if it's DNA formed, it should be thymine here. If it's RNA formed, it should be uracil. During copying, only difference is complementary to adenine, uracil nucleotide, uridine monophosphate should be attached instead of thymidine monophosphate. That is the only difference when RNA is copied from DNA. When DNA is copied from DNA, thymidine monophosphate should be added opposite to adenine. If it's RNA copied, uridine monophosphate should be added. All other places will be same. The enzyme is different. It is RNA polymerase enzyme. We will discuss it later. Anyway, the copying is like that. When this DNA forms an RNA, okay, suppose this is that RNA formed, is having this kind of base sequences. Okay, one just representation I have given. I mean, an RNA copied from this strand of DNA will be producing, suppose it is producing this type of an RNA strand. Okay. And similarly, suppose this strand also is copied to RNA. It is not taking place. Suppose this strand also is copied to RNA. Okay. This RNA produced will be, this from the opposite strand. So this RNA produced will be complementary to this. That means here will be uracil, here will be adenine, here will be guanine, here will be cytosine. Like this. Means the two RNAs produced from the two opposite strands of DNA must be complementary. They are not similar RNA. This RNA is not similar to this. These two are entirely complementary. Then what happens? Immediately they can pair. Okay. Such two complementary RNAs come together means they may pair and form double-stranded RNA. Okay. This is the problem. The first reason is when two strands of DNA are copied, okay, it will produce how many RNAs? It produces two RNAs. Okay, then what's the problem of producing two RNAs? These two RNAs are complementary. So what is the problem? They pair to form double-stranded RNA. Okay, a double-stranded RNA can never produce protein. For protein production, its bases should be exposed. We will discuss it. So, a double-stranded RNA cannot be translated. Okay, this is the first reason why the two strands are not copied to RNA. What is the first reason? If both the strands are copied, it produces two different RNAs which are complementary, immediately they can pair. They pair to form a double-stranded RNA. Being complementary, they pair. Okay, then what happens? The double-stranded RNA can never be translated. No protein production. Okay, the second possible reason put forth by scientists. If these are not pairing, okay, suppose, fortunately, these are not pairing, then also it is trouble. I mean, this RNA is not similar to this RNA. These two are two different RNAs. This will be translated to one protein. Okay, this will be translated to another protein, not similar protein. This will confuse all the things that are stored. Normally, you know that one portion of DNA is responsible for, that gene is responsible for one character. That means it is forming one RNA. That one RNA is producing single protein. This is the genetic information flow. This should be altered. Means if both the strands are not paired, okay, if they remain as two separate RNA, still it is problem. It will produce two different types of protein. That is impossible for one segment of DNA. Okay, that way the genetic information will be trouble. So what is the second reason? If both the strands are if both the strands are copied and it is producing two strands and they are remaining unpaired, means they are two different RNA strands. Okay, not paired means they remain as two different RNA strands and they copy to two different proteins. Okay, that may confuse the gene expression. Okay, from one portion of DNA, only one protein is required. That is the flow of genetic information. If it was the other way, it will confuse the flow of genetic information. Okay, is this part clear? Why the two strands of DNA are not copied? Only single strand is copied. Scientists put forth these two possible reasons. Okay, shall we move to the process of transcription? Okay, in transcription, as we have discussed already, certain regions of the DNA only undergoes transcription. These regions we can call as transcription units. Okay, the region where the transcription takes place is known as transcription unit. Then, what are the components of a transcription unit? Okay, so what is transcription unit? It is a segment of DNA, okay, where the transcription takes place. Okay, let me do that. One strand of DNA, the opposite strand of DNA. Opposite strand of DNA. Okay, this strand direction is 3 prime, 5 prime written, 5 prime to 3 prime it is written. Okay, listen, see here, these are the two segments, two strands of a DNA drawn. Okay, that means suppose this portion of the DNA is responsible for one production of a protein, whatever. If this is a transcription unit, it must have a sequence where the transcription begins. Okay, and it must have a sequence where transcription ends. Okay, you know the full length DNA is not a transcript. The region where transcription begins is known as promoter, and the region where transcription ends is known as terminator. Okay, promoter, terminator. Any doubt? So understand, in the transcription unit, where the transcription begins is called the promoter. There is no such box here. This is simple DNA only. We are marking this in a box. This is a promoter sequence. Then here is a terminator region where the transcription ends. That means from here to here only RNA is produced. That much long RNA only produced, not the entire long RNA. Okay, then see, when we tell we have already, when we discussed the first part, we have already discussed only one segment of the DNA is transcribed. That segment of DNA is called template DNA. Okay, template strand. Opposite strand is known as coding strand. It has no any great role here. Still it is known as coding strand. The opposite strand is. So understand the transcription unit. Here you can see the coding strand is not coding for anything, but it is taken as the reference strand. The direction of the coding strand is taken as the direction of this DNA. That means the DNA's 5' end is this one, because this is the coding strand's 5' end. This is the reference point. Okay, so which is the 5' end of the DNA means this is the 5' end. It is based on coding strand. Coding strand is the reference. The 3' end of the DNA is this side, because that is the 3' end of the coding strand. Any reference is based on coding strand. Okay, then listen here. See, towards the 5' end, the promoter is located, and towards the 3' end, the terminator is located. Okay, so understand this very carefully. If the coding strand and template strands are given, you can write which is promoter, which is terminator. Okay, any one if is given. Okay, suppose this way it is given. This is template strand. Given as, this one is given as coding strand. Okay, then coding strand's 5' end, what is promoter? Coding strand's 3' end, that is terminator. That means if any one of these strands is given, you can write which is promoter, which is terminator. Where is promoter, where is terminator. Similarly, if promoter and terminator are given, you can predict which is coding strand, which is terminate strand. That way, questions may be asked for your exam for 1 mark. Simple 1 mark question may be asked. Okay, so just understand, in a transcription unit is nothing but a DNA segment which is involved in transcription. Okay, which part of DNA is transcribed to an RNA? That part is known as transcription unit. It has a sequence where the transcription begins, that is known as promoter, and it has a sequence where the transcription ends, that is known as terminator. Okay, and the two strands, the strand which is copied is known as template strand. The strand which is not copied is known as coding strand. Coding strand is taken as the reference strand. So its 5' end is the 5' end of the DNA. So promoter is located towards this. Then coding strand's 3' end is the 3' end of the DNA. Terminator is located towards that end. Okay, this is template strand, this is coding strand. Already I have drawn, I think it is clear. Then one more thing, if this is promoter and this is terminator, the region between is known as structural gene. Okay, this is what actually is getting transcribed to RNA. This region is getting transcribed to RNA. Okay, so that region is known as structural gene. Keep in mind, we have to elaborate it later. Okay, so that is about the transcription unit. Then what is about it? Something more about the promoter sequence. This promoter. What is special about the promoter? Why at the promoter only the transcription begins? You know, transcription is carried out by which enzyme? RNA polymerase enzyme. Transcription means what process? Building of RNA. Polymerization of RNA based on DNA template. So the enzyme is a DNA dependent RNA polymerase. It is working dependent on DNA template. Then DNA dependent RNA polymerase is the enzyme required. See, this enzyme can access only the promoter sequence. What is the reason? There are definite binding sites for these enzymes here. One such example in Escherichia coli is pribnobox. Okay, pribnobox is a particular conserved sequence, a definite sequence present in the promoter. I mean, Escherichia coli promoter, within the promoter sequence, there is a region known as pribnobox. T. Okay, pribnobox means this is a sequence of this many bases. T. Okay, this sequence is present within the promoter sequence of Escherichia coli bacterium. Okay, its corresponding sequence will be there in the RNA polymerase enzyme. Okay, to bind it. So, RNA polymerase can only access this point because this point has certain conserved sequences like this. So, not there in your textbook, you will study in higher classes. Anyway, just understand for one conserved sequence in the promoter, this pribnobox. Okay, a conserved sequence within the promoter of an E. coli bacterium is pribnobox. Its sequence is this. Pribnobox means this, the sequence. It is present in this region. Okay, so such kind of boxes or conserved sequences make this binding specific. This enzyme can access only this point because of this type of reasons. Okay, so that is about the transcription unit. And at the transcription unit, which region is accessible to RNA polymerase? Where does the RNA polymerase go and bind? To the promoter region. Okay, which consists of definite conserved sequences like pribnobox. Okay, I think it's clear. One example just I have given, that only. Then one thing. Okay, DNA. If the template strand of DNA sequence is given, DNA template strand sequence is given to you. Okay, see, is given ATGZ like this is the sequence of DNA of template strand. Okay, template strand sequence is given. The question is, write down or identify the sequence of mRNA transcribed from this. Identify the sequence of mRNA transcribed from this. Very easy. Okay, should be complementary basis, should be written here. And if a coding strand is given also, you should know how to write. Then try to write it, all of you. If this is template strand, all of you write to your notebook. Now you write what is the sequence of mRNA. What is the sequence of mRNA produced from this template strand? How to write it? Here, mRNA produced from this template strand. Adenine will be forming what? Uracil. Then here will be adenine. Here will be, instead of guanine, will pair with what? Cytosine. This can be repeated. I have given repeated sequence for easiness. Okay, like this. Okay, that means if the template strand is given, you can write the mRNA sequence exactly complementary basis, 5' to 3'. Exactly complementary basis you write. Okay, sometimes in the entrance question or in the options, you may see this one from this to here. The answer may be, this may not be there in the options. Okay, what is the sequence of mRNA given? This option may not be there. Instead, the option will be like this, from here to here. GCAU. GCAU. Is there any difference? GCAU repeated. Is there any difference for this strand and this strand? No difference. See, here direction is 5' to 3', it is written. Here from 3 to 5, it is same. The strands are same. Sometimes the option may be like that here. So you have to be careful by looking the options. Okay, that means the mRNA can also be written like this, GCAU, GCAU, repeat, GCAU like this. So okay, I think. That means this both are same. Sometimes it can be written starting from 5' or otherwise it can be written from that side to this side also. We cannot say the DNA remain like this in a cell. If we are looking from this side, we read it like this. If we are looking from the other side, we read like that. Okay, so upstream, downstream concepts we will discuss later. Okay, anyway, understand if the template strand is given, the mRNA strand can be written very easily by simply writing what? The opposite complementary basis. Okay, suppose if is coding strand given, what is given to you is DNA coding strand. Then how will you write? How will you write the sequence of mRNA produced from that? Out of you try. How will you write? This is the coding strand given. How will you write? Will you first write template strand? Then you write mRNA. That is the correct procedure. You have to first write the opposite template strand. I'll just show you. It's not necessary actually, I'll tell you. See, this is coding strand. This is not copied. What is copied is template strand. So you have to first build the template strand. T-A, C-G, T-A, C-G, like the template strand will be. Then you have to write what? This is the template strand. Then you have to write what? It's mRNA. That means A, U, G, C. Okay, A, U, G, C, like that repeat. That means when if is coding strand given and you want to write the mRNA means, you can first write the template strand based on coding strand. Template strand is complementary to coding strand. Opposite basis, just write and do the template strand. If is okay, then it's mRNA you can write. Complementary to this. Adenine will pair with not thymine, uracil. That is only different. Then just listen to me. Is there any similarity with this newly formed mRNA and this coding strand? Coding strand and mRNA only difference is instead of thymine, uracil is. All other things are same. Okay, so you need not write any template strand here. Just to do instead of thymine, just to write uracil. Then okay, that means if is coding strand given, the answer is very simple to write. Instead of every thymine, you write what? Uracil. Remaining things are exactly same. This is the newly formed RNA. Why so? Both this mRNA and this coding strand is complementary to template strand. Okay. Both this RNA and this coding strand is complementary to the template strand. And only difference is in the RNA instead of thymine, uracil is there. This way we construct. Any more doubt means we will discuss it later. Okay?
Thank you.